Hey Amber, you want to hear something funny?
Sure.
I've got my eye on you.
That wasn't funny.
Okay, let me try again. What did the hippocampus say after the breakup?
What?
Thanks for the memories.
Wow. Well, let's get back on topic.
Yeah, we're not talking about the brain specifically, but this is CSI, the science revealed, and we're going to talk about muscle contraction. The first part of the skeletal muscle structure starts with the tendons, which are fibrous tissues that attach the muscle to bone. Next, the entire muscle is encased within the epimysium, the outermost membrane of the muscle, which is also a dense connective tissue. Within the muscle, there are fascicles which are surrounded by the perimysium, another layer of connective tissue. The fascicles are composed of muscle fibers surrounded by the endomysium, which then are made up of muscle fibers.
Within the muscle fibers, there are muscle cells that consist of myofibrils, which are further subdivided into sarcomeres. These are the fundamental contractile units of the muscles and play a critical role to muscle contraction. The sarcomere is composed of two main parts, actin, or thin filament, and myosin, the thick filament. The myosin has myosin heads that bind to the actin, which is made up of actin, troponin, and tropomyosin. On the actin, there are active sites where the myosin heads bind, but they are covered up by the tropomyosin. With the binding of calcium ions to something called troponin, they are able to be shifted to reveal this binding site. Furthermore, different regions of the sarcomere are labeled with different letters. The M line signifies the middle of the sarcomere, the Z line or disc that ends in separation of sarcomeres, the I band, that is the lighter portion containing only thin filaments, the H zone containing only thick filaments, and finally the A band in the middle where both thick and thin filaments are found.
With muscle contraction, everything starts with a signal from the brain. When muscles are at rest, tropomyosin blocks the active sites on the actin, which means myosin heads can't bind to them. This is when action potential comes to play. First, a stimulus causes the sodium channels in the motor neurons membrane to open, letting sodium ions flood into the cell. Next, once the threshold is reached, more ion channels open, leading to even more sodium ions into the cell, which is called depolarization. This means the inside of the cell is positively charged and the outside negatively. Next, repolarization occurs by closing the gated sodium channels and opening the potassium channels, which leads to the movement of potassium ions outside the membrane. The neuron eventually becomes hyperpolarized, causing the sodium potassium pump to start so the neuron's polarized state is normalized. With this pump, 3 sodium ions are pumped out and 2 potassium ions are moved into the neuron. This travels down the cell until it reaches the axon terminal.
At the axon terminal, the electrical signal transforms into a chemical one in the form of neurotransmitters, which are stored in the vesicles. These cross the synapse between the motor neuron and skeletal muscle receptors, which causes another action potential that travels down the sarcolemma through the T-tubules. As a result, a signal leads to the diffusion of calcium ions from the sarcoplasmic reticulum into the sarcoplasma, so it can be used for the muscle contraction process. The calcium binds to something called troponin, which controls tropomyosin, a protein that covers the actin's active sites, preventing it from binding with the myosin heads. We will further describe this process with the sliding filament theory, which is an explanation for this muscle contraction.
First, the myosin heads bind to ATP, which hydrolyzes to ADP and pi and changes its shape to a high energy configuration.
Next, the high energy configuration allows the myosin head to bind with the actin. After calcium ions bind with the troponin in order to shift the tropomyosin off of the actin's active sites, forming a cross bridge, which is the link between these two substances.
The myosin now relaxes to a low configuration but remains attached in this configuration, which is referred to as a power stroke. This brings actin towards the sarcomere center.
Finally, ATP is needed to break the cross bridge and relax the muscle, thus reverting back to the starting position. But what happens when an organism dies? Well, when an organism dies, all normal bodily processes cease, including the production of ATP. As previously described, ATP is vital to the action potential, signaling the start of muscle contraction, moving the myosin heads into a high energy configuration, the active transport of calcium ions into the sarcoplasmic reticulum, and the breaking of the cross bridge for muscle relaxation. Without cellular respiration, there is no ATP, which means none of these processes can normally occur. However, the muscles can utilize the leftover calcium ions when they leach into the sarcoplasm and reveal all of the active sites that the leftover ATP stored by the muscles can bind to. This means that the myosin heads will bind to the actin and cause an extreme muscle contraction, thus the stiffness of the organism's body characterized by rigor mortis. Also, since there is no more ATP, the cross-bridge obstruction that relaxes the muscles can't occur. Despite this, over time, the bacteria in the body consumes many parts of the organism, which eventually causes the body to go limp and deteriorate further.